Within the controlled environment of the sericulture laboratory, the sericulture activity was carried out with precision and care. Setup included proper ventilation system, air conditioning, humidity chamber, heaters, and ventilation fans with rearing space. The process began with preparation of feed. Castor leaves were collected from nearby sources and they were thoroughly washed and disinfected using potassium-based solution. The leaves which were collected were thoroughly dipped 2 to 3 times to ensure proper cleaning, and after dipping them they were gently shaken until all the tree plots fold, and the leaves were then dried under the sun until they were completely free of moisture, then laid flat on the trees. As the process continued, the trees were sprayed and dusted with disinfectant and slake lime in the clothes. The new trays were then lined with the new newspaper, and these newspaper were placed flat on the trees. Then after that, the larvae from the used trays were gently transferred to the new trays which have newly lined newspaper. After all this, they were transferred back to the rearing unit, and all the waste material was collected and then it was kept separately, which can be further used for another application such as the leaves and the residues, silkworms residues. Then the trays were taken outside, they were disinfected properly, and then they were kept in the sunlight for few minutes. After this, the feeding followed a structured pattern. Fresh leaves were again collected, cut into the approx sizes, and provided to the larva. For early stages, first and second instar, the leaves were cut into the small pieces of 0.5 centimeters, and they were placed gently over the larva, allowing them to feed comfortably. The rearing activity then moved to the different instar stages, moving to the first instar stages and the second instar stage. The second instar in a humidity chamber to maintain the humidity, and then the stages moved to the larvae. Second instar, third instar. The third instar were seen growing and feeding, then fourth instar were seen, and then finally moving to the fifth and the last stages of the instar. At the stage, the larvae were more of the yellow color, and then they were seen going into the molting stages. The molting stages were identified different ways, that they started going away from the feed and started rolling away from the feed and lifting their head up, which can be clearly seen as all the behavioral changes become very evident. These larvae show the signs of molting, and following these stages, these larvae were kept into the mountages. These mountages were of both plastic and artificial and traditional method. The green plastic mountage and the traditional mulberry dried leaves mountage were used in the lab. The leaves acted as a natural mountage. The larvae began spinning the cocoons, enclosing themselves into the fine silk threads. The cocoon gradually formed layer by layer until complete, and these completed cocoons were then kept into the separate tray, sometimes covered with the another tray to maintain the suitable conditions. Here, male and female were both identified, as male have more bushier antenna as compared to the female moth. Then these moths were allowed to pair in the controlled, undisturbed and the dark environment. A sheet was placed beneath them, so whenever the female lays egg, it just transferred on the sheets. They were allowed to pair undisturbedly, which resulting in female laying the eggs, making the completion of one life cycle. Thank you.